Shubha, please put your presentation on full screen.
Yes.
Okay, we are live now. We'll just wait for another 2 minutes and then we can go ahead. Okay. Hello, everyone, and welcome to the fifth session of Burgeon. Those who are attending our session for the first time, Burgeon is an online platform provided for youngsters to encourage them in the field of wildlife conservation and environment. Every Wednesday, we have these sessions online wherein one student speaks about his or her work in presence of subject expert of their respective field. So, without wasting much time, let's begin with our today's session. So, today we have Miss Shubha Kauthankar. She is botany student at Goa University who will be presenting her work on mushroom diversity. And I'm very pleased to introduce our today's guest, subject experts, Dr. Biswas. He is a lead scientist of product development and lifecycle management at Gaiagen Technologies Private Limited. A very warm welcome, sir. And we also have
Thank you.
Priya Sule, who is Vice President of Human Relations and Marketing Communications at Gaiagen Technologies Private Limited. She is also director and co-founder of Living Heritage Foundation. Welcome, Priya. Thanks for joining us today. First, I request Shubha to start with her presentation, and later on we will listen from our experts. So, Shubha, all the best, and platform is all yours. Please go ahead.
Thank you, ma'am. Am I audible to you? Hello.
Yes.
Yes, you're audible.
Good evening, everyone. Good evening, everyone. Myself, Shubha Kauthankar, student of Goa University, and today I'll be discussing about my journey into this unique world of mushroom. So, firstly, I would like to thank Aranya Environment Research Organization for giving me this platform. So, my journey started some few years ago during the time of my college when I was introduced first to the bird watching as a hobby through GBCN, that is Goa Bird Conservation Network. And the time I spent in this outdoor looking at the birds and learning about them gave me ample opportunities to see the surrounding organisms too. So, and driven by the curiosity, I began learning about these as well. So, it was on such a trip that my eyes caught a sight of a beautiful tiny mushroom which looked like a tiny pink umbrella. So, at the beginning, all I wanted to know is its identity. And the very next day, I poured over some books, and using keys, I could able to identify the pretty pink mushroom. So it was later during my final year of MSc, I got to learn more in detail about the mushroom when I opted for mushroom biotechnology paper, which was taught by Dr. Nandakumar Kamath sir. So under his guidance, I got to know how detailed research has been carried out in this field of mycology. So moving further, most of us are familiar with the delicate mushroom species that raises their head from the soil during the rainy season. So these forms are the unique group of organisms in the kingdom fungi. So most of these disappear in few days or some within few weeks. What is visible to our naked eye is the mushroom, and the other form of fungi is basically the fruits or the fruiting body of the variable species. These mushrooms have a very interesting association with its host organism in the environment. And Western Ghats, with its moist and mild climate, is one of the world's most beautiful mushroom habitat, home to 1,000 plus species of fungi. So in Goa itself, there are more than 500 species of mushroom found. So just step off the trail among trees and shrubs. Monsoon is very ideal for this mushroom if you are looking for mushroom, and you'll surrounded by various varieties of mushrooms. So these are the books which I referred in the beginning just to identify the mushroom at least at the genus level. So the next question that comes in minds of many people is whether mushroom are plants or animals. So the answer is no. Fungi inhabit their own separate biological kingdom. They are neither plants nor animals. What makes them different from these two is the way they obtain their nourishment. Hence, they are also known as saprotrophs. Understanding the structure and life cycle of mushroom is prerequisite and help me a lot before starting up any work in this field of mycology. So basically, the mushroom is the fruit or the fruiting body that carries spores to ensure the continuation of its own species. So the upper portion which you can see here is the pileus or the cap, which consists of the reproductive organs or the gills, which produces the spores. Then the middle portion is the stalk or the stipe, which is attached to the central center of the cap. And the base is the mycelial thread, just like the root of a tree, usually unseen and present underground. So next, coming to the its life cycle, most of the mushroom have common life cycle. Mature fruit body disperses its spores, that is the reproductive structure, by various dispersal mechanism, either by beings or insects. These spores are favorable under the favorable condition like moisture or the temperature. They germinate, forming network of minute thread called as hyphae. So basically, these hyphae are of two mating types, positive and negative. So when two compatible hyphae meets, it results into formation of another network of thread-like structure called as mycelium. So this is the structure of mycelium. If you see carefully surrounding most of the tree or just underground where the mushroom has been grown out, you'll notice such network of threads thread-like structure. So the mycelium is like the root of a tree, which absorbs the nutrients from its host and helps the fungus to expand. So basically, the fruiting body or the mushroom which we have which we have seen growing in a garden, is nothing but the expansion of this mycelium. And similarly, the cycle continues. So coming to its habit, mushroom are classified into two major habits. That is the tree inhabiting mushroom and soil inhabiting mushroom. So as mentioned in the beginning, mushroom are host specific. They rely on other organisms, that is either plants or animals, for their nutrition source. Understanding habitats of mushrooms help me a lot in identification. So basically, mushroom inhabits dead barks of trees, and they grow in the soil. This is where I started my journey, my own village in search of these tiny little umbrellas. So, being a resident of my own village, I am really lucky enough to explore its biodiversity and could able to study minute details of the nature. So, the next question is how to identify a mushroom. So, unfortunately, mushrooms are very delicate things. That is, they do not last for a long time, and this makes researching them very difficult. So since we do not know where they are exactly found until they are in the fruiting stage, we get only to study it once, that is seasonally. So I tried marking the spot they are fruiting so that it can be observed and studied next time. So it was quite good idea, but mushrooms are funny things. So one can see a mushroom on a patch of land at 1 year, but it may not be seen again at the same area the another year, or maybe find completely different kind of mushroom in the same area. So, while studying mushroom biotechnology during my MSc final year course, I got to know the exact methodology how to identify a mushroom systematically. So, as taught by Dr. Kamath sir, it is mandatory to take spore print followed by studying its macroscopic as well as microscopic characters, followed by preserving it and drying it for the further use. Then, spore print is basically the pile of many spores. So once the spore print is appeared on a slide, it is examined under the microscope, and the description is then compared with the data already published on the field guides and other notes. So some mushroom can be also identified morphologically by examining their outer appearance itself on the field. So many people ask me this question when they find a mushroom, like, aren't they poisonous? So yes, some mushrooms are very poisonous, but never attempt to eat a wild mushroom without being 100% sure of its identity. Then coming to its function, um, luckily, I got a chance to work with some research students at Cholla Ghat. So during the fieldwork at Cholla Ghats, I could able to notice that most of the fallen wooden logs were associated with some kind of fungi. So basically, it absorbs nutrients from its host and thus are main recyclers in nature. So they break down woods and other forest litter to simple organic material. It amazes me how these delicate little things have the ability to completely break down even the biggest tree. Sometimes the tree can be associated with different fungi too. Say if a tree log is attacked by one species, say a oyster or a pleurotus species, it will slowly decompose the tree until it reaches a stage of decomposition that is no longer suitable for its existence. Then the same tree log or the bark will take over by some other fungal species, say a polypore. This succession will continue until there is no more tree or the bark is left. So the reason why they are called saprophytes due to is their feeding habits. Not all mushrooms on wood, though some species feeds on humus and organic material contained in the soil. So at the same time, they form a beneficial relationship with live trees also, and this is known as symbiotic relationship. Here, the tree provides with carbon source to the mushroom, and in return, mushroom gives essential minerals and exchange of nutrients. Now coming to its diversity, I have photographed and studied different species of fungi by observing its common field characters during various field trips and treks. So coming to the agarics, this is a type of mushroom which is characterized by the presence of a pileus, that is a cap, and clearly differentiated stalk, that is the which are gilled mushroom comprises, and it comprises the very large group under the mushroom family. So in this way, the gill attachment to the stem is an important character for separating species and genera. Coming to the hypoloma species, these are the fleshy agarics with tough cap. Surface is a little bit slimy and umbonate, usually found in cluster on the below the bark of the tree. Then the Melanoleuca species is a kind of agarics wherein the cap is usually convex and depressed from the top. Then Marasmius and Mycinus species. So these are the commonly found species you can easily locate in your garden. And I used to find it difficult to differentiate between these two, that is Marasmius and this Mycinus species. So I wasn't really aware of the sculptural quality of the mushrooms. So until I started noticing the tiny things based on the shape, of the cap and width of the stem and other general impression on the mushroom. So Marasmius has the typically have the long stipe and convex cap, highly variable gill attachment. The stipe or the stem is darkened in color and the gills are widely placed. Whereas the Mycena species have a conical cap and basically the short stipe. Coming to the next Lapiotoid family, this constitute the Macrolapiota and Lapiota species. So this usually have whitish to creamish fruiting body. They have free gills, basically decomposers, and mostly found on soils. These are quite similar to Amanita mushroom, which is considered as world's most poisonous mushroom. The next is hygrocype species, so mostly found in the forest floor, where which receives the highly rainfall. This picture I captured while trekking to Mollem Hill. The fruiting body is basically smooth and often found in many colors. This is Omphaluto species. The fruiting body is mostly dry, somewhat fragile, and have deeply decurrent gills. And cap is umbonate, that is, it has grooved at the center. And these are also decomposers of litter and wood. At the left hand side, you can see the Cortinaria species. So if you take a closer look of such mushroom, you'll notice that surface is basically wrinkled, pitted and cracked, and glutinous too, covered with slime. And at the right hand side, it is a Cantarellus species, easy to identify based upon its structures. So the fruiting body is usually the funnel shaped with smooth veined and the central stalk. Next is the Coprinus species, which usually grows on animal dung. So recognized by it is basically recognized by its free gills. Then it is found abundantly growing on dung and feces. The cap is scaly mostly and becomes black on maturation. And if you notice the gills, these are placed distantly. The next is the Pleurotus species, that is also known as oyster mushrooms. So they are the agarics, which are in a fan or a tongue shaped, usually lacks lateral stipe. So some species have stipe, which is attached on the backside of the gill, forming a radiating pattern, as you can see in this picture. So most of the species grows on woods, and some species are edible. Next is the puffballs or the gasteroids fungi. Uh, uh, The spores are produced inside the fruiting body, that is the onion shaped. At the right hand side, you can see the scleroderma species, the ball like structure. Um, so the spores are produced inside this fruiting body, which after maturation burst and releases the spores. So most of the species are ball shaped and mostly without stem. Some have star-like appearance. The left-hand side picture, that is the geostrum species, that is also known as earth star. Um, they are hygroscopic, that is, they open only when they come in contact with water. So this is a cup fungi, Cookenia species. This constitute the family of Sarcoscyphaceae, and you can able to locate this species on fallen branches, trunks, etc. The fungus have deep cup-shaped to funnel-shaped fruiting body. The inner spore bearing surface is brightly colored, and this color is fades usually after drying. So it has hair-like structure present inside the cup, made up of hyphae. So in this picture, you can see the stink horns. So the other day, my sister told me, Didi, there is some type of egg in our parking area, and some tail-like structure is there. So I was amazed, like how come an egg have a tail-like structure? So go to see, it was a bulb of a stinkhorn species. So I just later I kept it aside in a safer side, and next day it was hatched into a beautiful stinkhorn, the one which you can see in this slide. So the fungus has slimy entirely and the dark spore mass at the top of it. So at the same time, the fruiting body emits some nauseating smell which attracted the ants and some other flies. So with the help of magnifying glass, I could able to notice some ants carrying the upper portion of the cap, that is the black spores. Maybe the reason could be dispersal of the spores. This picture I captured at Cholla Ghats, and the right hand side is the another species of stinkhorns, that is the Clathrus species, also called as the bracket stinkhorn, cause of its cage-like appearance and especially found on the soil. So the fruiting body is irregular and oval to egg-shaped. This is usually found alone in the leaf litter and on the garden soil. Next is the bracket fungi or the polypores. So these are the unique group of fungi found in cluster or sometime in solitary. Together it constitute the polypores. One such type of fungi forms tubes inside the fruiting body, and the most of the species are wood decayers. So the pores or the tube mouth, which you can see in this picture, the tubes other side of the fungi can be spherical or the gill-like. So the fruiting bodies can be completely attached to the host of the substrate. As you can see in this picture, this is a species of Didaleopsis. So it have well-developed caps, as you can see in this picture. So rarely there is also stem, and it can be soft and annual, tough to hard. In some cases, the perennial also. So this is a microporous species. A perennial fruiting bodies can be recognized by typically having several tube-like layers and broad zones of the cap. So all polypores are basically decomposers, and these perennial fruit bodies continue to produce new tube layers. The growing periods can be often seen as the raised concentric zones on the cap. This is the Ganoderma species, which is used in the medicinal application. These are the trumets. Next is the Auricularia species. So genus of jelly fungus, also known as the wood ear. The surface of this fungus is usually slimy and found growing on both living as well as dead tree. The fruit body is variable in color and shape. Next is the Dacrymyces species. You will notice it on usually on palms, coconut, etc. So usually identify its orange, yellow, gelatinous to rubbery fruit bodies. This is the Claveria species, also known as the coral fungus, usually thread-like, and coral fungus usually with the upright orientation found growing on dead trunks. These are the Xyleria species, also called as dead man's fingers, as almost most of the species are darker in color. So most of these are inconspicuous species because it is erect and fruiting bodies grow in cluster at the base of the tree. Coming to the next, Goan Termitomyces species. So these are mainly known as the wild mushrooms, or in Konkani it is known as ormi. It grows mostly on the places known as termite mounds or the ruin. Interacting with elder people in my village, I got to know that ruin is constructed by usually by the termites, by most fertile soil from different places, and these include even small pieces of leaves and litter. So the main purpose of this termite mound is a store. Is the use for storage of food collected by the termites. So the most of the time, the ant hill are occupied by snakes and other organisms too. So once they leave this termite mound, only leftover material by the inhabitants of the ruin remains. So all these things facilitate the growth of special kind of fungus, which we known called as termitomyces. It forms a kind of fungal mass, also called as ormo or the fungal comb, and gets matured when it get proper humidity, moisture, and environmental conditions created inside the ruin or the termite mound. So once the fungal mound is matured, it produces mushrooms which we called as Roinichiormi. So, but if the anthill or the termite mound is disturbed, the fungal comb gets damaged and never get matured. So, termitomyces, these are some termitomyces species which I came across during the time of fieldwork. So, Tomatomyces is basically a monsoon delicacy in Goa and often brought into market by foragers from the village. Basically, these people are the only living link into the ancient forest around us. So there has been certain threats to Goan Termitomyces species. One of the reason is overexploitation. Some people around, without any proper knowledge of its life cycle, harvest the mushroom at very early budding stage, which inhibits the next cycle. The other reason is increase in the growing rate and destruction of the habitats due to construction work. So this is the guide which I used to identify the mushroom in the beginning. These are the references. And at last, I would like to thank Dr. Kamath for the whatever guidance that he gave during the course of this study. My friends and my family for the support, and thanks again to Aranya Environment Research Organization for giving me platform to share my work. Thank you.
Thanks, Shubha. It was a really nice presentation, and I'm very happy to listen from you because mushrooms is one of the subject which have been ignored by people, and we don't know many things about mushrooms. So it was nice to see there are so many mushrooms which are around us, but we tend to ignore them, right? So it was really great. So before, let's move ahead and ask Dr. Biswas, like, what does he think about your presentation, and what is his feedback?
Mhm. So overall, the presentation is actually quite good for anybody who is, interested to know more about this domain of science, and particularly for identification. Uh, Morphology is to some extent can get you only so far. You can identify certain taxa in certain cases maximum up to general level. But if you really want to identify to the species level, what you should do at least is look through a microscope, particularly the spores. Huh? So sculpture on the spore, how it looks like, even the size. For certain mushroom species, everything else will be identical except for the size of the mature spore. That's the only differentiating factor between two species. And now we also know that there are even more different species that is genetically distinguishable from one another, but morphologically speaking, they are identical. You cannot distinguish them between from one and another. So, if you really want to explore and kind of keep a record of whatever you are observing, so one part, the basic part is just you can classify up to family level, even genus level, just on the field. But if you want to go ahead with a species level identification, at least you should look through the microscope, and the best thing would be to go ahead with the ITS sequencing. So ITS is something that is a part of the genome between 5.8s, 18s, and 28s. So between these RNA genes, you have little bit of string of DNA. Those are not really part of the RNA gene, uh, but that is the part you will get enough variation, but not too much variation. So that is a standard for identifying any fungi, not just mushroom, any fungi, based on ITS sequencing. So that is the ultimate classification criteria for mushrooms. But anyhow, doesn't matter if you're looking to classify or identify till species level or general level. So when you are presenting something so diverse, now all those genera and all, a fantastic way to represent that would be uh, create a tree. Hmm. So these are the species belonging to this genera. This genera will belong to this family. This family will belong to this order. But anyhow, most of the mushrooms they are only basidiomycota only. But still, you can have a family order. Most of them are also aggregates as well. But still, you will have at least family level diversity, order level diversity. So just counting species is absolutely fine for documentation purpose. But also having this additional information will help you understand. Uh, See, classification it represents phylogeny, uh, and phylogeny means evolution. So how these organisms actually evolved, so that can be understood only by creating phylogenetic trees. If you have ITS sequence, of course you can do a lot better, phylogeny study. But even if without ITS, if you can identify even up to general level, still you will have some diversity at family level, order level. So that will also reflect in the overall abundance and diversity of mushrooms in the region that you are surveying. And one more thing is that there is something fundamentally very different between termitomyces and all other mushrooms. Hmm. And that part, I mean, for your next presentation, maybe you can elaborate a little bit on the difference, because the way you presented to new people who are just trying to look into mycology, it won't they won't feel the difference between a termitomyces and all other mushrooms. So, and that is also one part you can highlight. For termitomyces, you see, ultimately, all living things on Earth, doesn't matter if it is a mushroom or anything else, everything is about getting access to resources. Hmm. Now, for mushrooms, for all mushrooms and all fungi, of course, they get their nutrition from already prepared nutrition by others with help of plants and animals, everything. So they don't really synthesize anything on their own. They accumulate and then they just rearrange the molecules to suit their requirement, basically. But even if they are doing just that, still, Termitomyces lost some of their actually majority of their abilities to survive on its own. And that happened only once in the entire evolutionary history of fungi. It happened only once that termites and mushrooms they form an alliance. And once it happened, it diversified from there, but neither the mushroom nor the termites could ever get back to a free living condition ever. So this mutualism it happens only one time, and it never was reversed during the entire history of evolution of either termites or fungi. And if you just think of it, ultimately it's all about gaining access to resources. Hmm. For example, it's not all organisms will have all the metabolic capabilities to utilize resources of all kind. So, for example, these termitomyces and these termites, all both of them together, they can use cellulose, hemicellulose, the most uh, how to say robust of biomaterial that is very difficult to break down and utilize properly. So what happened for termitomyces is they have certain specialized role to degrade one part of the entire reaction from, let's say, cellulose hemicellulose to glucose or maybe just acetyl CoA. So this pathway, one part termitomyces they perform it a lot better than anything else. And then there is another part that lives inside the gut of termites. So, and those organisms, they do it some part of that entire reaction better than anybody else. So this is a kind of tripartite coalition. So whenever you are getting the raw material, cellulose, hemicellulose, but something that is very difficult to digest, it passes through the termite gut. So it get partially digested kind of way. And the remaining digestion, Termitomyces do. Now, for Termitomyces, they are doing it very well, but they have lost most of the other vital capabilities that will help them to survive on their own. So when you remove the termite comb from the mound, both termite as well as Termitomyces, they both perishes. If you keep the comb inside the termite inside the mound, just remove the termites. Still, the Termitomyces cannot, flourish in that comb without the termites. So they need constant grooming, and the entire gut microbiome of the termites, they also evolved through time by acquiring different protists, flagellates, then just losing them, gaining bacteria. So it's a very complex gut microbiome that is present inside the termites. So along with that gut microbiome and termitomyces, both they utilize cellulose, hemicellulose as best as possible. And by allowing that, Termitomyces, they're providing shelter, uh, arborage, grooming, everything. They're cleaning it up, the comb and Termitomyces. And by doing that, Termitomyces, they are able to establish in the comb, grow in mycelial state, then come up with a fruiting body. And the pinheads that grow on these combs inside the mound, the termites also consume them. So, as in any alliance, they're both getting out of something from this alliance. But the problem is both of them have lost their ability to live without one another. So, in that sense, Termitomyces is entirely different from all other mushrooms. That is one thing you may consider highlighting a little bit. Apart from that, whenever you're documenting, taking photographs of specimens in the habitat, try to use some kind of scaling. You can either keep one currency, I mean, one coin, or you can use a graph paper, or scale, something to let the audience know about the actual size of the fruiting bodies. So you can take a photograph of microcarpus that will look like 6 feet. Uh, but nobody will know the exact size, and sometimes that is something is required whenever you are going back to the field, or someone who is using your presentation as a kind of reference to look for mushroom in the field. So try to include a reference for size. And whenever possible, try to take photographs of the underside of the mushroom. Take one from top, one from the side, one from the underneath, and particularly uh, keep in the background at least one any kind of scaling system. If you don't have anything, definitely you will have at least couple of coins, na, currency, 1 rupee, 5 rupee, whatever it is. Just place it by the side of your sample, take the photograph. But if you are planning for a field visit, then carry some scale, graph paper, anything. One thing that actually is very simple that you can make yourself. There are microscope slides, na, glass slides. You can take two glass slide and paste one graph paper inside that glass slide, then cut it out. So even in the rainy condition or whatever the condition is, you will have a scale. So you can always place that glass slide by the side of your sample and document it. So yes, it looks like you have covered a lot of area nearby and got good coverage of what is available. And one thing, this is just something I wanted to ask you. While studying Termitomyces, uh, just approximately how many species of Termitomyces you actually have encountered? There are record of around 19, approximately 19 in that region, Goa and all the Western Ghats region. But how many actually you have encountered? I'm just curious.
Shubha, can you answer the question? Are you able to hear us?
Hello.
Yeah, Shubha, how many Termitomyces species that you have encountered when you were studying?
Hello.
Hello, hello, Shubha.
Hello.
Yeah,
Uh,
how many species?
yes,
Uh,
yes, continue.
yeah, how many species of Termitomyces that you have encountered during your study, sir, is asking.
Hello. There has been
Uh,
some network issues, I guess.
right, right.
Uh, yeah, sir, I think she has some network issues, but I'm sure
Mhm.
she will be able to answer it shortly when she gets connection.
Yes. Okay. Anything
Yeah,
else?
yeah. Sir, you want to add something, or shall I
No,
go
that's all.
ahead?
That's all. That's all. We can take
Yeah.
up questions.
Yes. Thank you so much, sir. Actually, in fact, have said will encourage our student to take up this field because we didn't know that there are so many opportunities and so many options which are beyond taxonomy which we study in our books, right?
Mhm.
And there are so many techniques that student can opt for, right from DNA sequence to studying genetic material, right? And at least I had never thought about studying mushroom at genetic level, so I'm
Mhm.
sure that there might be many students who might take up this in future. Right. So I think before taking up question, I would request Priya to give her feedback
Mhm.
first, and then we can take up questions from the public.
Mhm.
Um, Hi. So I won't do too much of feedback also because Shubha is coming and going, and I'm not sure if she can hear everything. But just to, hey Shubha, hi, can you hear us? No, no, not yet. Okay. Well, I don't know if you can
No,
hear us
ma'am.
too. I think you're on a bit of a delay. I don't think you can hear us too well, but I felt like your excitement was really visible in everything that you were sharing, and you have a very strong personal connect to what you're doing. So that is also very amazing. I like that it has
Thank
started
you,
from
ma'am.
because that means it's going to go somewhere. Um, yeah, I did kind of. I felt like somewhere towards the end, you were trying to say that. Uh, just correct me if I'm wrong, but what I understood was that you said that tomatomyces would grow on an abandoned mound. Was that right? On an abandoned termite mound? I'm just not sure if that was what she mentioned. I was a little confused about that. But okay, it's okay. We can have this part of the conversation. I don't think Ashita, I don't think she can quite hear our questions too well.
Yeah, yeah. I think she has some network connectivity at her end, but I'm sure that she will answer your questions when she gets network connection.
Yeah, apart from that, I think more importantly, as feedback, I would say it's really amazing that you guys at Aranya are doing this. And it's
Thank
really
you.
important to do this because just in this last conversation, there was like four very strong, concrete bits of feedback about how someone can really get up to the next professional level. And I think that academia does amazingly well to inspire you, but then it's professionals who show you where things can go in the future. And it's amazing that you're putting those two together, and hats off to you guys for doing that. That's a great
Thank
initiative.
you. Thank you, Priya. So I will. Since Shubha has lost her connection, there are quite a couple of questions which have come specifically for Shubha, but
Just
I
if
hope
you
she will
can
be
make
able
a
to
note.
join us.
Sorry, just if you could make a tiny note for the quest. For one of the things as feedback for Shubha was just that she's so clear on the reason why there's this decline in population. It would be
Right,
amazing for her to actually partner with someone and to communicate back to everyone, maybe a method that can help restore balance. Because I think mycology's most amazing gifts, which we haven't enjoyed as yet, is restoration. And
right, absolutely.
it starts with communicating things effectively. So from a marketing point of view, I think communication could save a further decline in the wild population, and since she knows a lot, she must go out and communicate that.
Yes, definitely, Priya. And this presentation will be there for her to see it again, because I'm sure that she will see it again, like because she has lost the connection. So
Yeah,
your feedback she will be able to hear. So
I'm glad.
yeah, let's go ahead and take up the questions that have come. So first question is by Emmy. You sorry if I have pronounced your name wrongly, Antonio. Is there any prospect of study of chemical compounds present in psilocybin mushrooms for treatment of leading mental disorders like depression and anxiety? Uh, so
This is
if you
from you.
can take up.
Oh, okay, yes. Of course, there are, and people have been working on this field for the last 25, even 30 years. And with improved capabilities like HPLC, GCMS, and also standard, clinical trial regimens, we're also getting some, how to say, substantial evidence that they do work. And the one problem is Mm, several mushrooms, they look so close to one another, but they differ in their genetic makeup. So whenever somebody is doing one study based on just the morphological features, and it actually has been like this at least before 2004. So sometimes what happens whenever you refer to some publication and you see one species, let's say species X, it does have one, two, three properties, and the key of science is reproducibility. So if someone is getting some particular results or phenotypes on species X, another one anywhere from the world should be able to get the same result if using the same species of mushroom. But the problem that occurred is people were relying mostly on morphological features. So lot of times misidentification did happen. So whenever somebody says that, well, they found some positive effect of particular mushroom without a clinical setup, basically, and when somebody looked into it, they couldn't find it. But that was before 2004. But mainly after 2004, due to the reduction in cost of sequencing and all, now people are getting the identifications more accurate. And after 2015 or 2013, most of the papers, whatever is information is presented, it will match. If you get the same species and perform the same study, you will get the same kind of results. But particularly for psilocybins, uh, people have been trying to generate enough evidence for the last more than 20-25 years. But until very recently, we got some evidence, but still it's just in infancy. Oh, so there is plenty of scope to work on psilocybin. And two key aspect will be: first, to get the compound properly identified, and two, to get a proper clinical trial design, so that it's like a double blind placebo kind of study. So you, first of all, for mental disorder, measuring mental disorder is itself very difficult. You have to be able to quantify. Just for example, let's say MDD, major depressive disorder. There are different way to measure it with different questionnaire, and sometimes it's not very accurate. Hmm. And so to be able to see effect of the compound, you should be able to measure it properly. So in that sense, that's one of the reason it's a little bit difficult to generate enough evidence, or how to say, if the effect is substantial, that will reflect definitely. Hmm. But for effects that are more of a kind, how to say, subjective, so it's difficult to generate evidence for that kind of study. So you need a way to measure the disorder that you are trying to treat. You need to identify the compound properly, and while designing the study, it should be placebo controlled, double blind. So the person who is taking the medicine should not know if it is a medicine or a placebo. The doctor administering the drug will also not know if it is a placebo or a real compound. It will be matched after even getting the study analyzed, not even before the analysis. So analysis will be done based on the randomized code. After the analysis is complete, then only look into basically a proper clinical trial. Hmm. And you need to identify the compound, uh, and also you need a way to measure the mental disorder. Even professional psychiatrists and psychologists, we don't have any concrete measure to particularly to measure most of the mental disorders. For something that has physical representation, that is easier. If you have fever, you can measure it with a thermometer. If you have a tumor, you can get a scan. You will know about it. But for mental disorder, till now it's very difficult to measure. I actually had some experience working with one Danish company for uh, medicines that work against some specific mental disorders. So at that point, I had to go through a lot of clinical trial studies and how clinicians actually measure disorder. So particularly, I know for this particular case, psilocybin and mental disorder, uh, evidence is there, uh, but it may not satisfy everyone. That's what I'm saying.
Okay. All right. So let's, thank you, sir. Let's move to next question. Okay. Krishnakant Bakshi asks, which atmospheric parameters mostly affect the growth of mushroom and fungi?
Mhm. First of all, they need a source of nutrition. They cannot grow on their own. Second, most of them need high humidity, high relative humidity. And third is the temperature. So for humidity, it's quite similar across different mushrooms. But for the choice of substrate, that is diverse among groups. And also for the temperature, that is also variable among groups. And apart from temperature, moisture, and nutrition source, another aspect will be electricity, static electric discharge. Actually, particularly in Japan, whenever farmers had lightning, striking their fields, no, they used to predict that they will have a greater crop of mushrooms, and that was just one observation. But now, just in the last 5 7 years, we have demonstrated clearly in the lab that for certain species of mushroom, certain voltage discharge through the substrate can increase the fruiting body production up to 200%. That is twofold. It can double the production. And also there are people who actually designed some funny devices. It's like two set of wheels connected by a circuitry, and you have inoculated some place substrate, and you know where it will grow. So you take the wheel, so two wheels will work as two electrodes. You just uh, drag two wheels through the substrate, and it will fruit a lot better than without electricity. So we don't know yet about all the parameters. What all we know is you need nutrition, you need temperature, and you need moisture. And but usually temperature is lower, moisture is higher. Usually, but it's not universal thing. Apart from that, there could be many variables that we are not aware of yet. Another point was, apart from electricity, uh, people also observed that uh, certain kind of natural phenomenon can have effect on fruiting. For example, if you have a forest fire, and if you don't have a forest fire, and you look in the next season from the same area, the area with forest fire and the area without the forest fire will have different uh, diversity of mushrooms popping out. So, and that is mostly due to the ash. The wood it burns, the ash it drips down through the rain. So, mineral. So again, there will be an effect of minerals electricity. So those things we don't understand very well so far. All we know for sure, and we can predict very well based on temperature, moisture, and nutrient source that we know very well. And to some extent, for certain species, we also know specifics about the pH of the nutrition source, the media. Apart from that, you have mineral content, you have static electric discharge, even high frequency sound wave. So what happens is when sound travels to the substrate, for some mycelia, it becomes too much. So the mycelia just break. It breaks open the mycelia. So when they do, they secrete some of their internal cytoplasmic content. That signal is picked up by the remaining intact mycelia around that broken mycelia. So they start to come up with sporulation and fruiting bodies. So lot of variables are there. It's very difficult to list all the parameters.
Right. Thank you, sir. I hope, Krishnakant, you have got your answer. So, moving to Priya, I do have one question for you, since you are into management part. How does a student of biology, especially like zoology or botany student, proceed to find career options in corporate sectors which deals with biology and related work?
Wow, you asked me a really difficult one, Harshada. But yeah, I mean, I feel like there are so many disciplines and there are so many alternatives at the moment. I mean, interestingly, so in a very present and current scenario, yes, there are a lot of corporates like organic chemistries can do anything from designing perfume to designing pheromones. So it really depends on what your second line of inclination is. You know, if you're particularly directed towards say conservation or reduction of use of chemicals, so there are options there. Similarly, I think microbiology and mycology is I feel like it's something that's still unleashing itself. It's not even arrived, you know, it's still in the process because more and more you start learning that there is so much more that can be done which does not require even in just in sustainable agriculture. So many options right now for microbiologists or entomologists, for a lot of people who are coming straight out of the sciences to actually contribute and in fact to assess and to establish impact of one against the other and use a proper scientific approach and kind of also undo maybe a lot of things that we've done in the past and to offer alternatives to them. um, I think that the real fun is going to be in the future of all of this because the truth is, look at everything around us, and it's becoming so clear that restoration is very soon going to be a paid occupation. And as unfortunate as it is, it's also a great
It
opportunity.
already is actually.
It is, it is. It's really coming down to the fact that I mean now that you see it in action and you start seeing how it's growing, restoring everything from the earth to your mind is heading in that direction. So
True.
um, yeah, I believe that the truth is everyone who's doing these disciplines right now are on the brink of a whole new reality, and it's a reality that they have a lot to contribute towards, and it's the perfect time to actually adopt the sciences while also in your mind adopting a very multidisciplinary approach. It's no longer the time when mycology had its own, sort of structure to it and its own discipline and did not include, say, another discipline like entomology. Shubha's presentation today is about the fact that there's a termite teaming up with a mushroom, and maybe it's about that entomologist teams up with a microbiologist. So yeah,
Right.
I
That's
think
true.
my approaches to restoring all the damage that our race might have done so far, it's going to be a well-paying job, and uh, to everyone who's taken that step already.
Yes. Thank you, Priya. So I would like to ask question to Biswas sir actually. Sir, how corporate sector can contribute or help in generating information about poorly known species like that of mushrooms, for example?
Sorry, your voice got a little bit, jumbled up. Can you repeat the question, please?
Yeah, yeah. So, I was asking how corporate sector can contribute or help in generating information about poorly known species like that of mushrooms.
Okay. So, uh, for exploration of mushroom, basically there are numerous ways. Yeah. Uh, One way is to generate, synthesize active pharmaceutical ingredients. Yeah. So, for those kind of work, molecular identification is absolutely must. There are some work that involves generating material like uh, alternative for leather, alternative for just fly ash brick. You can have a block made out of fungi that is light enough to float, harder than normal brick, and also fire retardant. So those kind of new material you can come up with mushrooms, and also leather, similar texture to leather. So these kind of things, it's okay for them to just get enough information about the species, but they need not get a proper molecular identification. When you are going and looking into for mushrooms that are produced for food usage, uh, for traditional usage, people do not really bother much about identification. They just do it from their past experience, and that is fine as long as you are consuming, harvesting and consuming on your own. When you want to do it for a commercial scale, whatever strain you are multiplying, it should be properly identified. Uh, Most of the companies still rely on getting the spawn from a source like culture collection. So there are different bodies who keep and preserve the store these spawns, the starter culture, and they can get the culture from the collection. There are very few companies who actually have their own culture collection. So if you are talking about just uh, growing it in a commercial scale and bringing it to the market, you don't really have to have the ability to identify it thoroughly. You will just get it from a source. MTCC, ATCC, their type cultures are available all over the world. And if you want to grow something that you collected yourself, then it's a must that it should be properly identified. And for apart from this study, basically you have material, you have active pharmaceutical ingredients, you have food source, and another part is actually waste management. And that is the part you are least bothered about the proper identification. If it works, it works. But for food grade, if you are getting it from some other source, it's already identified. And for pharma, it must be identified up to the strain level with proper molecular barcoding using RTS. So
Okay.
identification is one part that I would say the most basic part of the entire research. Finding applications to the available knowledge, that is even more important. So if you have use of one particular fungus, you can get it identified very easily. It's not that difficult. So what I would say for in general mycology, uh, the potential for a career using any aspect of mycology, that is dubbed as a white technology just because of the color of the mycelia, or mycotechnology. So you can have environmental restoration work, bioremediation work. You can have agricultural impacts like uh, entomopathogenic fungi. You can have a phytotonic effect using fungi. You can have mycorrhizal fungi to support plant growth. You can come up with molecules with medicinal property. You can have material like leather and brick. Basically, you can use fungi for everything you want. And one thing we often underestimate, because it's not that much visible. The bakery goods, the yeast, it's also fungi, and the brewery wine, it's also due to fungi. So we have been using fungi knowingly or unknowingly for thousands of years. The point I would like to make, it's not just for mycology. For any young scientist and students, the most important thing you can learn is how to learn new things. So you will never be able to learn everything, that is guaranteed. But the best thing you can learn during your academic career is how to learn new things. So whenever you face a problem, you can always find a solution. So that is the most important skill that you can have. And apart from that, it's important to choose certain techniques and tools that you would like to master. You cannot master all, but you should choose certain techniques and tools uh, that you can perform without any questions and with as much accuracy as possible. And to be very fair, for mycology, as diverse its applications are, tools and techniques used in the field is also very diverse. It's not just limited to mycology anymore. So you have to learn about analytical chemistry, GCMS, HPLC. You have to learn about even physics. You need to come up with a tensile strength of leathers and bricks, fire retardant. So you have to learn about physics. You have to learn about chemistry, and you cannot get masters or PhD on all. So you need to learn how to learn new things. That's the most important thing to know.
Right.
And yes, it's a exciting time to venture into the field of mycology. And to be honest and fair, it always has been. Huh. It's not like we're just speaking now, so it's happening now. It has been happening for a very long time. Huh.
Right. So I think we have to move ahead from like just going in a straight line of taxonomy or something. We have to explore more and then go ahead with in the respective field that we are interested in. So the
Hmm.
idea of thinking should be changed. Like this doesn't end at the taxonomy level, but there are a lot of options to look forward to. Right. So
Taxonomy is just the beginning of
right.
doing anything. Hmm.
Right, sir. Yeah. So, my last question actually is for Priya. Priya, do you think that is it necessary to have synergy between corporates and academicians or academics that has been done?
Yeah, actually, that's what I commended you on right at the start as well, because I do think that I wouldn't even say corporates anymore, because I think that, like I said, a lot of things are changing, and it's you're going through almost careers in that sense. You have like an academic career where you build on a certain kind of information, and what happens with your professors, especially the really good ones that really inspire you, is that sort of they give you like that little bit of fuel to start up on something, and they nourish like a certain interest within you. But I feel like it's really important, right, as frequently as possible during your educative phase, to be able to go out and do more projects. Like if you can take everything that you're doing maybe for the first 6 months and go out and apply it for 6 months and then come back and do something, and then go out again, then that's definitely a much better way of learning, because there's practice happening constantly. And I think that's also where corporates come in, where projects come in, where yeah, you get the opportunity to go out and try something before you finish 3 years and have to suddenly focus on figuring out if you actually acquired any skills through those 3 years. So I
Right.
feel like that's something that could change with education. Industrial training could be something that happens periodically. Students get to sit and learn different things. Like so, 6 months in college, 6 months on field, 6 months in college, 6 months on field, and suddenly I feel like you will see a massive acceleration in how skills are acquired. So definitely a good way to ally. And yeah.
Right. Right. Well
Peer
said.
reviewing is something that you guys have just put in place right now, where you have peers sitting down and looking at each other's presentations and asking questions and stuff like that. And it becomes much easier to accept that once you get into the actual discipline as a scientist, because then that becomes a great way of giving and taking knowledge with each other. So yeah.
Oh, right, absolutely correct, Priya, and that is the reason why we have called you on this platform today, just to have a broader idea, like moving ahead from whatever the classical taxonomy field that we do, right? Yeah, so I think we are done with the question and answer session. And Shubha is not on the stream right now. I think she has got some internet connectivity issue. But whenever she comes back, there are a few questions which have come for Shubha. But I hope she will be able to answer them in the comment session when she gets proper internet. So I take this opportunity to thank Dr. Biswas and Priya for joining us today, and it was really a wonderful discussion that we had. And I hope that our younger generation from Goa can collaborate with you all for like doing future studies in their respective field of interest. And we look forward for collaborative work in future as well. Yeah.
So do we.
So yes. So to end with the session, I would like to announce that for next Wednesday we will have Samruddhi, who will be speaking about her experience on working in a nursery and a poultry, and she's also a snake rescuer. So it would be also nice to have you all on the stream once again. Right. So thank you everyone. So with this, I announce that this session is we are ending this particular session. Thank you so much, sir. Thank you, Priya, and thank you, Shubha.